Hi, I'm Miss Prabhavati SN. In today's module, I would be explaining about the biosynthesis and oxidation of fatty acids. Let us begin with the introduction. All cells in the plant produce fatty acids. Their synthesis must be well controlled to balance the supply and demand for acyl chains. They do not exist free in nature. Primarily, they exist as unbranched hydrocarbon chains with an even number of carbon, which exerts variable saturation with hydrogen. Fatty acids are basically classified depending on the number of carbons, the number of double bonds and the position of the first double bond in the chain. The chain length and degree of saturation determines the melting temperature of a fat. Fats with shorter fatty acid chains or with more double bonds remain liquid at room temperature. Fats containing long chains remain solid at room temperature. Oxidation of fatty acid is considered as one of the critical metabolic pathways. The oxidation of fatty acids yields acetyl-CoA, which is the major energy yielding pathway in animals. For instance, the complete combustion or oxidation of palmitic acid is known to yield 2.380 kilocalories per mole. This energy is particularly important for cardiac and skeletal muscle. The products of beta oxidation can also be utilized by number of other tissues, especially the liver, kidney, small intestine and white adipose tissue for the production of ketone bodies. This in turn could be utilized as a source of energy. This is critically dependent on tissue, nutritional status, exercise, infection and various other pathological conditions. The mechanism of fatty acid oxidation was discovered by Franz Knoop in 1904. He found that the process involves the degradation of fatty acids into two carbon atoms. The process of combustion of fatty acid into carbon dioxide and water is reported to occur in mitochondria. Now let us understand the mechanism involved in the biosynthesis of fatty acid. The major site for fatty acid biosynthesis is the cytosol, and this process essentially requires sufficient concentration of NADPH and acetyl-CoA. The pentose phosphate pathway in the cytosol generates NADPH. The malic enzyme helps to oxidize malate into pyruvate and carbon dioxide, resulting in the generation of NADPH. The major building blocks for fatty acid biosynthesis are the acetyl-CoA molecules, which needs to be activated in order for the fatty acid biosynthesis to take place. The major building blocks for fatty acid biosynthesis are the acetyl-CoA molecules, which needs to be activated in order for the fatty acid biosynthesis to take place. Acetyl-CoA undergoes carboxylation to yield malonyl-CoA through the biotin containing enzyme acetyl-CoA carboxylase. This carboxylation reaction is irreversible and is the first major step in the fatty acid biosynthesis. The carboxylase reaction is similar to that of pyruvate carboxylase and propionyl-CoA carboxylase. For the activation of bicarbonate, ATP is used in the form of carboxyphosphate and results in the carboxylation of biotin molecule. Upon activation, the carbon dioxide group is transferred to acetyl-CoA to produce malonyl-CoA. Acetyl-CoA carboxylase consists of mainly three domains: a carrier protein of biotin carboxyl group, biotin carboxylase that adds carbon dioxide to biotin, and a trans carboxyl group which facilitates the transfer of carbon dioxide from biotin to acetyl-CoA to yield malonyl-CoA. It is been postulated that there exists a allosteric mechanism for the regulation of acetyl-CoA carboxylase. The animal acetyl-CoA carboxylase is a filamentous polymer which consists of protomers. Each protomer is known to have biotin containing carboxyl carrier protein, carboxylase and the trans carboxylase domains along with allosteric regulatory sites. The enzyme is most active in its polymeric form, whereas the individual protomers are not. The overall activity of acetyl-CoA carboxylase is highly dependent on the equilibrium between the two forms of the enzyme, that is inactive protomer and active polymer. Citrate serves as a precursor for the formation of acetyl-CoA in the cytosol, which also acts as an allosteric activator for this enzyme. The phosphorylation of acetyl-CoA carboxylase modulates the regulatory effects of citrate and palmitoyl-CoA. This mechanism is assisted by protein kinase and protein phosphatases, which lead to dephosphorylation. The palmitoyl-CoA would show a preference for binding to phosphorylated enzyme. A very small amount of palmitoyl-CoA is needed to inactivate this. The intermediates generated during the fatty acid synthesis are not linked to coenzyme A. Rather, they would get attached to an acyl carrier protein. The next step is the fatty acid elongation, which initiates the process of formation of acetyl ACP and malonyl ACP. Both acetyl trans-acylase as well as malonyl trans-acylase catalyzes the reaction. Once acetyl CaAs and malonyl ACP have been formed, the chain elongation process begins. The subsequent reactions are the reverse of beta oxidation, where ketone is reduced to the alcohol by the enzyme beta-ketoacyl ACP reductase. In the next round, the butyryl ACP is transferred to sulfhydryl group of beta-ketoacyl ACP synthase. The decarboxylation of the second molecule of malonyl-CoA generates a nucleophile which attacks the carbonyl thioester of the butyryl S-CaAs to form C6-beta-ketoacyl ACP. This is later reduced by beta-hydroxyacyl ACP, which undergoes dehydration to form C6-delta-square-enoyl ACP. This is then reduced to form C6-acyl ACP and is transferred to the sulfhydryl group of beta-ketoacyl ACP synthase, which tends to condense with third malonyl-CoA until it forms palmitoyl ACP. In the next step, the thioesterase hydrolyzes the thioester bond of palmitoyl ACP to generate palmitate and ACP. Fatty acids are hydrophobic in nature and are extremely insoluble in water. The triacyl glycerols would get segregated into lipid droplets. They must be emulsified before being digested by water-soluble enzymes in the intestine. Once absorbed, they must be carried in blood by proteins, which helps to counteract their insoluble nature. The carbon-carbon bonds in a fatty acid are relatively stable, which can be overcome by the activation of carboxyl group at C1 position by attaching with the coenzyme. This allows for stepwise oxidation of fatty acyl groups to take place at C3 position. This carbon atom is referred to as beta, and hence the oxidation of fatty acid gets the name beta oxidation. Both liver as well as mitochondria exerts an unusual property, that is, they are unable to oxidize fatty acids or fatty acyl CoAs unless carnitine is present in sufficient amount, which promotes catalytic action. Free fatty acids or fatty acyl CoAs are not capable of penetrating the inner membranes of liver and other tissues of mitochondria. Acyl carnitine can easily penetrate the membrane and is converted to acetyl CoA within the matrix. Now let us discuss the major reactions involved in the oxidation of fatty acids. The free fatty acid which enters the cytosol from blood is unable to pass directly through the mitochondrial membrane. It must undergo a series of enzymatic reactions involving a three-step process. Step 1. The first step is initiated by catalytic action from a group of isoenzyme acyl-CoA synthetases that are present in the outer mitochondrial membrane. The cell comprises three different types of acyl-CoA synthetases, which mainly acts on fatty acids of short, intermediate and long chain respectively. One type may be involved in activating acetate and propionate to its corresponding thioesters. Activation of medium chain fatty acids from C4 to C11 is initiated by another form of synthetases, while the third type is exclusively involved in the activation of fatty acids from C10 to C20. The enzyme acetyl-CoA synthetase catalyzes the formation of thioester linkage between the fatty acid carboxyl group and the thiol group of coenzyme A to produce fatty acyl CoA. Acetyl-CoA is a high-energy compound. Their hydrolysis to free fatty acid is known to produce negative standard free energy change. The process of formation of fatty acyl CoAs becomes highly favorable by inducing hydrolysis of two high-energy bonds in ATP. The inorganic pyrophosphatase enzyme immediately hydrolyzes the pyrophosphate, which is obtained by the activation reaction. This is known to precede the activation reaction, leading to the formation of fatty acyl CoA. Step 2. The outer acyl-CoA esters formed in the outer mitochondria are not capable of crossing the membrane. Further, fatty acyl group oxidizes and is transiently attached to the hydroxyl group of carnitine, while the fatty acyl carnitine with the help of carnitine acyl transferase 1 catalyzes transesterification of fatty acyl group from coenzyme A into carnitine. Through the facilitated diffusion mechanism, the fatty acyl carnitine ester crosses the inner mitochondrial membrane to enter the matrix with the help of acyl carnitine or the carnitine transporter. Step 3. This is considered as the final step. In this step, the carnitine acyl transferase 2 facilitates the enzymatic transfer of fatty acyl group from carnitine to intramitochondrial coenzyme, which is located on the inner surface of the inner mitochondrial membrane, which helps for the regeneration of fatty acyl-CoA, which is then released along with free carnitine into the matrix. This carnitine further re-enters the space between inner and outer mitochondrial membrane. This mechanism is assisted by an enzyme, acyl carnitine or carnitine transporter. This fatty acyl-CoA, as it gains access into the internal surface of mitochondria, is finally ready to undergo oxidation with the help of a set of enzymes present within the mitochondrial matrix. The entire process comprises 4 basic steps. The first stage is a reaction that is catalyzed by 4 enzymes. In the initial stages, the dehydrogenation process of acyl-CoA produces a double bond between alpha and beta carbon atoms that produces a delta square enoyl-CoA. Further, the first step is catalyzed by 3 isoenzymes of acyl-CoA dehydrogenase. Each one is known to act based on fatty acyl chain length. For instance, very long chain acyl-CoA dehydrogenase mainly acts on the fatty acids which contain 12 to 18 carbons. Medium chain fatty acyl-CoA dehydrogenase generally acts on fatty acids carrying 4 to 14 carbons, fatty acids having 4 to 14 carbons, and short chain acyl-CoA dehydrogenase generally acts on fatty acids carrying 4 to 8 carbons. All these enzymes are basically flavoproteins which also contains flavin adenine dinucleotide as its prosthetic group. The electrons that are removed from fatty acyl-CoA is then transferred to FAD. The dehydrogenase, upon being converted into its reduced form, immediately donates electrons to an electron carrier in the mitochondrial respiratory chain. The oxidation reaction is catalyzed by an acyl-CoA dehydrogenase, which is analogous to succinate dehydrogenase in the citric acid cycle. In both the reactions, the enzyme would bind with the inner membrane and a double bond is introduced into the carboxylic acid between alpha and beta carbons. FAD basically acts as an electron acceptor. Ultimately, the electrons from the above reaction enter the respiratory chain, which is then passed on to oxygen. This process results in the concomitant synthesis of 1.5 ATP molecules per pair of electrons. The second step is a beta oxidation cycle where water is added to the double bond of the delta square enoyl-CoA to produce L-stereoisomer of beta hydroxyacyl-CoA. The major catalyzer of this reaction is enoyl-CoA hydratase. In the third step, a dehydrogenation reaction takes place which converts L-beta hydroxyacyl-CoA to form beta ketoacyl-CoA with the help of the enzyme beta hydroxyacyl-CoA dehydrogenase. Here, in this step, the major electron acceptor is the NAD+. Further reactions tend to produce NADH, which donates electrons to NADH dehydrogenase. The basic function of this enzyme is to carry electrons to the respiratory chain. This in turn produces ATP from ADP as electrons pass into the oxygen. The reaction that is catalyzed by beta hydroxyacyl-CoA dehydrogenase is reported to be an analogous process much closer to the malate dehydrogenase reaction observed in the citric acid cycle. The last and the fourth step of beta oxidation cycle is catalyzed by acyl-CoA acetyl transferase commonly known as thiolate. This acts as a promoter for the reaction to take place between beta-keto-acyl-CoA with a molecule of free coenzyme A. This results in the splitting of the carboxyl terminal 2 carbon fragments of the original fatty acids as acetyl-CoA. The other products produced due to the reaction is the coenzyme A thioester of fatty acid, known to be shortened by 2 carbon atoms. This particular process is termed as thiolysis. The last 3 steps of the sequences are reported to be catalyzed by either of the 2 sets of enzymes. The type of enzyme that may be involved is in turn dependent on the fatty acyl chain. For the fatty acyl chains having 12 or more carbons, the reactions are mainly catalyzed by a mechanism involving multi-enzyme complex that are basically associated with the inner mitochondrial membrane, referred as trifunctional proteins. This TFP is a heterooctamer consisting of alpha 4 beta 4 subunit. Each alpha subunit is known to comprise 2 activities, namely the enoyl-CoA hydratase and the beta hydroxyacyl-CoA dehydrogenase. The beta subunits are known to contain the thiolase activity. These 3 enzymes are known to have very strong association, which further allows for efficient channeling of the substrate from one active site to another without causing diffusion of the intermediates from the surface of the enzyme. The trifunctional proteins would bring about the shortening of fatty acyl chain either to 12 or to a very few carbons. The further oxidation reactions require 4 set of soluble enzymes in the matrix for the catalytic reaction. The single bonds of methylene groups present in fatty acids are considered to be relatively stable. The sequence of reactions taking place in beta oxidation process could be an elegant process for destabilizing and breaking of the highly stable bonds. The first 3 reactions are known to produce less stable carbon-carbon bonds. The 4 steps of beta oxidation are repeated in order to yield acetyl-CoA and ATP. In one cycle of beta oxidation process, 1 molecule of acetyl-CoA, 2 pairs of electrons and 4 protons are eliminated from the long chain fatty acyl-CoA. In other words, a shortening of 2 carbon atoms would occur. Upon removal of 1 acetyl-CoA from palmitoyl-CoA, the thioester enzyme A from the shortened fatty acid still remains in the matrix. The resulting myristoyl-CoA then passes through another 4 set of beta oxidation reaction to provide second molecule of acetyl-CoA and lauryl-CoA. On the whole, the process involves 7 beta oxidation sequences to oxidize 1 molecule of palmitoyl-CoA to yield 8 molecules of acetyl-CoA. The successive oxidation reaction produces FADH2, which donates a pair of electrons to ETF of the respiratory chain. This yields 1.5 molecules of ATP. Each molecule of NADH also delivers a pair of electrons to the mitochondrial NADH dehydrogenase. Its subsequent transfer to oxygen produces 2.5 molecules of ATP. Thus, it results in the production of 4 molecules of ATP for each carbon unit. Water is also produced in the course of reaction. For hibernating animals, the oxidation process serves to provide metabolic energy, heat and water for animals that do not eat or drink for very long periods. The palmitoyl-CoA undergoes subsequent oxidation process to yield 8 acetyl-CoA molecules. This acetyl-CoA could be further oxidized to yield carbon dioxide and water by the citric acid cycle. Now let us move on to discuss the disorders associated with the fatty acid oxidation. The storage form of triacyl glycerols serve as the chief source of energy, which is utilized for muscle contraction. An inability to oxidize fatty acids from triacyl glycerols is known to result in serious consequences. The major effects resulting due to certain defects in fatty acid metabolism have been discussed below. First is the defects resulting due to carnitine uptake. It is considered as a genetic defect of plasma membrane which leads to primary carnitine deficiency. The common manifestations during the first year of life includes fasting intolerance, hypoglycemia and sudden death. In older patients, it is generally manifested as progressive cardiomyopathy and muscle weakness. The condition can be promptly treated by taking carnitine supplements. Next is the defect in mitochondrial fatty acid transport. It results in a condition called carnitine palmitoyl transferase 1 deficiency. In some cases, it leads to the development of fatal hepatic encephalopathy and myopathy. Next one is the deficiency of acyl carnitine transferase. It causes chronic progressive liver failure, non-reversible hyperammonemia. The other common features are skeletal myopathy and very rarely hypoglycemia may be seen. Medium chain acyl-CoA dehydrogenase deficiency. It is one of the most frequently encountered disorders of fatty acid oxidation pathway which results in the occurrence of inborn errors of metabolism. Patients with this disorder appear to be normal at birth, though they may be having the presence of transient episodes of benign form of hypoglycemia. Between the ages of 3 and 24 months, acute decompensation sets in as a result of prolonged fasting and infections which may impair usual food intake. Due to the metabolic decompensation, it may lead to unexpected death. The metabolic stress may quickly progress towards the development of life-threatening situation. Now let us summarize today's episode. All cells in the plant produce fatty acids. Their synthesis must be well controlled to balance the supply and demand for acyl chains. Oxidation of fatty acid is considered as one of the critical metabolic pathways. The oxidation of fatty acids yields acetyl-CoA, which is the major energy yielding pathway in animals. The major site for fatty acid biosynthesis is the cytosol, and this process essentially requires sufficient concentration of NADPH and acetyl-CoA. The free fatty acid which enters the cytosol from blood is unable to pass directly through the mitochondrial membrane. It must undergo a series of enzymatic reactions. An inability to oxidize fatty acids from triacylglycerols is known to result in serious consequences.